Yeah, I'm now recording. Yeah. So we have attendees joining in. Hi, Blake. I'll just send you a message.
Ah.
I think we can start, or wait for more people. Yeah. Hi, Blake. So I'm going to start. So, hello everyone. So welcome to this week's Polariton chemistry webinar. So, before we start, I will show some results. As you can see that here is the future schedule that next week we will have Stephanie Rich from Fabry University Berlin. And we will also begin the roundtable discussion of the ground state vibronic polaritonic chemistry, and more details coming soon. And Dr. Blake Simpkins will be the panelist of it. And remember that our Polariton chemistry webinar is held every Wednesday at 9 a.m. Pacific Standard Time. And for each week, you must register to participate through this link, or we will send the reminder email. And you can register through the link in the reminder email. And also, since we recently created the Polariton Chemistry online community Facebook page, so feel free to post your papers or maybe some interesting stuff or your advertisement there regarding this community. And also, we have created the Polariton Chemistry webinars YouTube page. And for those who missed the this talk, we will upload some of them, and you can watch it later. And also, let's begin to introduce some of the webinar mechanism. So during the talk, you can raise your you can click this raise hand button to raise your questions, or you and I will interrupt the panelists at appropriate time and unmute yourself to let you ask questions. And or you can use this chat function or maybe this Q&A function, so that some of the questions will be answered after the talk. And here comes to our today's talk. It is our honor to have Dr. Blake Simpkins from U.S. Naval Research Laboratory, and to give this today's Polariton webinar talk. And the topic will be vibrational cavity coupling, modulation of coupling strengths and ground state reaction rates. And now let's begin. And Blake, you can share your screen now.
Okay. Hello. I assume you can hear me. So my name is Blake Simpkins. I'm from the Naval Research Lab. Of course, I want to thank Wei and Joel and the other members that are helping organize this for putting this together. It's been fantastic, keeping us engaged during the quarantine and maybe even engaged a little bit more and interacting more than we were before the quarantine. And I hope everyone's doing well out there. I'll be candid and tell you that it's been pretty taxing emotionally on us out here, but we are all healthy, so we've got that, and I hope everyone else is doing okay. So today I'm going to talk about our work focusing on vibrational strong coupling. And I guess in case somebody got lost and found themselves in this seminar by accident and don't know what strong coupling is, I'll tell you how I define it and how we think about it in the context of our work. So if we begin with an optical resonator that exhibits some resonant transmissive or reflection response, and load within that optical resonator a material that has a very strong optically active transition, those two oscillators can exchange energy, maybe exchange energy a few times before it decays, resulting in the creation of two new hybrid light matter modes called polaritons. In energy space, it kind of looks something like this. We have a cavity oscillator, a material oscillator. They interact and form two new modes. This picture looks quite a bit like bonding and anti-bonding orbitals of a diatomic molecule. The coupling strength is termed the Rabi splitting, and that goes as the transition dipole of the material, or in the case of ensemble coupling, which is what we look at, that goes as the square root of the concentration of material absorbers in the cavity. Okay, so this is nice. Why do we care about all this? This is why. It turns out that the presence of these new hybrid states can have profound effects on system properties. They can facilitate energy decay and energy transfer. These new states can allow co-coupled elements to exchange energy with one another in new ways, and alter fundamental properties such as work function and conductivity of semiconducting materials. And most excitingly and interesting to me, and probably many here, is the prospect of altering chemical reactivity or chemical processes. And so I highlight in this panel down here the work of the Evison group, who have been essentially creating and defining this field, at least the experimental aspect of it over the last number of years. Their first example of cavity modified reactivity, I believe, is shown here, and that was a dye isomerization reaction, which they slowed under excitonic strong coupling. And then more recently, they've released maybe a half a dozen or even a dozen reports of vibrational strong coupling to impact chemical reactivity. And this figure here is taken from their probably most important publication, their science work, which shows a reaction rate that is cavity tuning dependent, that slows when certain vibrational bands are coupled to a cavity, and even more excitingly, a variation in the resulting product branching ratio. Okay, and so this tells us that not only can we monkey around with reaction rates, but we might be able to impart some unique selectivity to chemical reactions. Okay, and this is what we're pursuing in our group, is modified chemical reactivity. So what I'll tell you about today is organized here. I'll start with our very basic exploration of system properties and how they impact strong coupling. Our work on trying to apply electrochemical actuation to strongly coupled systems. Our first attempt at modifying a chemical reaction within a cavity. And if I have time, I might spend 30 seconds or so just highlighting a couple of a bit of the ultrafast work my collaborators do over here. Okay, so let's start. So about 5 years ago or so, we published our first work on vibrational strong coupling. This was about the same time the Evison group also published. Both works center on Fabry-Pérot cavities coupled to a carbonyl bond within a polymer thin film. We were very excited about that. Completely unbeknownst to us, several years earlier, Dan Wasserman's group had accidentally, according to him, coupled a metamaterial or metasurface resonance with a phonon mode in a silica spacer layer. So they had been writing about and describing strong coupling to phonons, much earlier than us. And of course, strong coupling between oscillators has been a concept that's been around for quite a while. But translating that to molecular vibrations and certainly applying it to chemistry was new. And so we kind of hit the ground and started running with it. So any physical property that's modified via cavity coupling, let's say reactivity, relaxation kinetics, etc., likely the most important factor influencing those processes will be the magnitude of the Rabi splitting. So one would want to control the Rabi splitting, and one of the easiest ways or direct ways to do that might be to, within the ensemble coupling process, would be to change the concentration of the molecular absorber within your cavity. So we grabbed this molecule, hexamethyl diisocyanide, which has two strong isocyanate bands, vibrational bands, and we blended that into PMMA and spun thin films. And what we found was that when we increase the concentration of that absorber, the Rabi splitting increased. It scaled with the square root of that concentration. That wasn't much of a surprise. This square root scaling for ensemble coupling had been seen and analytically described for a couple of decades before we came aboard. But we felt it was important, number one, for us to understand what we were doing and what we were seeing, and to begin to explore what sorts of behaviors translate from the excitonic coupling field to vibrational strong coupling. And coming from, well, some of us were coming from a field of spectroscopy expertise. This relationship, the square root dependence, was a bit counterintuitive. So what this tells you is that the interaction between this ensemble of molecules and the cavity does not depend on the absolute absorption, but on the absorption per volume, okay? And this was a bit counterintuitive, but to verify this fact, and again, to kind of teach us and make us more familiar with how this type of behavior works, we created a number of cavities of different thicknesses and loaded them all with a solution of the same molecular concentration. So the absolute absorption of those molecules increases, it scales up. However, the interaction with the cavity, the splitting, remains about the same. Okay, and that's shown here. So these are path length. This is the Rabi splitting is a path length independent response, at least within this range. This experiment was also important for us because this was, early on, and we wanted to be absolutely sure we were seeing this strong coupling phenomenon and not some spectroscopic artifact. So specifically, you might argue that, well, rather than creating two new polariton states, you simply have a single Fabry-Pérot transmission resonance, and the absorption of these molecules is carving out this big dip in the middle and making it appear that you have these two polaritons. If that were the case, as you increase the path length, increase the total absorption, that carving out artifact would get larger and your apparent splitting would increase. But in fact, it doesn't. So this is just another verification to us about what sort of phenomena we were seeing. Okay, so this property, Rabi splitting, is path length independent. So path length doesn't matter for it. However, path length does matter for certain other system responses. And this was some very exciting work based on measurements Adam Dunkelberger did at the Naval Research Lab. So here we see a number of transmission spectra taken at different probe fluences. Okay, and what you see is that at low probing fluence in dark blue, we see our two polariton transmission peaks, as we expect. But as you probe this system with higher and higher optical fluxes, those two polariton peaks contract a bit, they approach one another, and eventually at high enough fluences, the resulting transmission spectrum looks like a single peaked function, similar to the bare Fabry-Pérot transmission peak. And what's going on is that as you illuminate this cavity and photons bounce back and forth within the cavity, eventually leaking out and being detected, they have a chance to excite some of the molecular species in the cavity. And when they do that, there's a ground state bleach. You now have a reduced difference in population between the ground state and first excited state, a weaker absorption within that molecular layer, and therefore the coupling is weaker. And that's what we see here. Note, this is an optical modulator, okay, an intensity-dependent optical transmission. If you're interested in that sort of thing, you can measure the fractional transmission as a function of power, which we do here. You also want to know a key parameter of such a device, which is the saturation fluence. So the saturation fluence is the optical flux needed to reduce an absorber's absorption by a factor of 2. That is an intrinsic property of a material. Okay, so it doesn't matter what the concentration of the absorber is or the path length. That saturation fluence is just a function of the extinction coefficient, essentially. And so you measure or you extract the saturation fluence with plots like this. So if you want to know the absorption as a function of incident fluence, that's also proportional to the ground state population as a function of fluence, which is proportional to the Rabi splitting squared, right? So we measure the Rabi splitting as a function of incident fluence, fit it to this expression, and extract saturation fluences. Now, I do want to point out that although I don't think saturation fluence was calculated, this Rabi splitting saturation or contraction has been seen before in the excitonic field in these two publications down here. And I want to apologize / thank Matt Pelton for his contribution here. I was not aware of that until I saw his presentation in this seminar series a few weeks ago. Okay, so we extract saturation fluence as a function of system properties. Now remember, this is an intrinsic property of a material. However, under the strong coupling conditions, we're able to impart some tunability, okay, to this parameter. And what we see is that as the concentration of molecular absorbers increases, or the Rabi splitting increases, the saturation fluence increases. So it takes more photons to excite the same number of molecules, essentially. And there's also a dependence on cavity length. What we think is going on qualitatively, or with a picture, is shown here. So the absorption spectrum of the molecules shown in this dotted line, and under strong coupling, the system exhibits these two polaritonic peaks. Those resonant transmission features act as windows through which incident photons sample the molecular absorption. So as the Rabi splitting increases, these polaritonic windows scoot further toward the wings of the molecular absorption, and excitation of the molecules becomes less efficient, right? And that's why the saturation fluence increases. Furthermore, an increased cavity path length causes these polaritonic windows to narrow, and that just narrows your window that you're able to access the molecular absorption by. So strong coupling it can be very important in this optical limiting scheme, because it gives you a means by which you can tune an otherwise intrinsic property. Okay, so continuing along with the theme of using system design to control coupling strength, we imagined a Fabry-Pérot cavity and a single slab of molecules that will be coupled to the cavity. And we figured, well, if you place that slab at different positions within the cavity, one could explore the impact of molecule optical mode overlap and how that influences coupling strength. And intuitively, you expect that the coupling strength is maximized at the peak of that mode, but let's see if we can learn something new. Okay, so we built such samples using spinning techniques, and we placed a PMMA slab at different positions within the cavity. We saw that as the slab was moved toward the center of the cavity, the splitting increased, and that's plotted here. So this blue shows the cavity mode profile for half the cavity, and in red we see that the data follows that profile. Coupling is strongest at the antinode and weakest when the molecules are at the mirror face. We also did a bit of analytical modeling. These open squares are an analytical model that essentially just integrates the molecular absorption over a cosine squared function, which describes the mode profile. Okay, so this isn't that exciting necessarily, but we did learn something new by making a mistake, of course. I guess that's often how you learn. This type of cavity supports a p-polarized and s-polarized mode. Qualitatively, they have the same shape. However, the p-polarized mode is non-zero at the mirror surface. And so when interrogating this same sample under p-polarized conditions, you measure a different Rabi splitting. That's completely understood, and we can describe it with knowledge of these various modes. That's fine. But it reminds us that strong coupling is occurring whether photons are present or not, right? This is interactions between the vacuum fields and the molecular oscillators. So when designing a control cavity, you ought to make sure that there are not optical modes that can couple to your molecule that you are unaware of. You're simply blind to them because of your interrogation geometry, okay? And we played one more little trick with this system that I think is interesting. So if we just focus on these. Oh, yeah.
Yeah, so we have one question, so.
Sure.
Okay. Sorry to interrupt.
Hi, so I have a question about the last slide that you just showed that you were moving. Yes. So for this kind of cavity, you always have the other modes, right? So the third mode or the second mode. So when you move it, wouldn't you see many maximas instead of just seeing a maxima of Rabi splitting in between?
So let's see, I assume you're talking about the higher order modes.
Yes, yes.
So yeah, they are present as well, but they are at different frequencies. And in fact, I don't have the slide, but if you look at the paper, we make use of the second order mode as well. So the second order mode is at roughly, I don't know, 2400, 2500 wave numbers or something like that. That second order mode spatially does exhibit two maxima, yes. It occurs at this higher frequency, 2400 wave numbers, which also coincides with another vibrational band. And so actually using this same set of samples, we inspected that second order band interacting with that other vibrational mode, and the coupling strength there followed this second order mode profile as you'd expect.
I see. Thank you.
Yeah.
We have one more question. Sorry.
Yeah. Yeah, that's okay.
Hey Blake,
Yeah.
nice talk so far. I like that your comments about this dependence on the polarization, the coupling strength on the polarization. But for most of the experiments, as you mentioned, that they are done in the dark, which do not really have a polarization dependence, right? So how should I understand that?
Yeah. So that's sure.
Sure.
Well, oh, go ahead. Is there more part of the question? Okay.
Yeah, no, that's all my
Okay.
question. Yeah.
Yeah, so that's right. So but remember or imagine that the coupling is occurring whether photons are present or not. So even when you're not interrogating the system, there is interaction between the vacuum fields of the cavity and the molecular excitation, right?
Right.
So we use light to try and see what's happening. But even if we don't use light, even if there's no photons, there's still interactions going on. And if those interactions are causing distortion of the potential energy surfaces or this or that, that's going on whether there's photons present or not. So if in this case there happen to be two different modes that are present, one polarized one way and one polarized another way, whatever I imagine whatever interactions mode A is having with the molecule and mode B is having with the molecule, they are both active, they're both happening. And if you measure some physical response from the system, if those two different modes are influencing the physical response of the system, you'll see the impact of both of them. Okay. So for instance, in this case, we see that, yeah, coupling is pretty weak when you put this slab of molecules near the mirror. Okay. And let's say, for instance, we were able to selectively measure the reaction rate of this slab of molecules, right? They're at the mirror surface. Coupling appears to be really weak. You would expect no impact on reactivity, for instance. Oh, but wait, there is another mode supported there that we're just not looking at that's imparting quite a bit stronger coupling. And so maybe that other mode is influencing the chemical reaction rate or some other observable. So
I see. Yeah. I see. That's a great point. So we really need to consider all the photonic mode.
Yeah, that's right. All the photonic modes, whether you're illuminating them or not. Yeah.
Thank you.
Sure. Okay. So then briefly here, so if you look at these blue slabs, this is just a slab of molecules we placed in the cavity. We did that in the previous slide. It gives us some coupling strength. Now we add a second slab at some different position in the cavity. Okay, so now we have two slabs. And we see that when we add that second spatially separated slab, this coupling strength increases. It increases by square root of 2. Yeah, that's simply because we've doubled the concentration of molecules in the cavity. Not that big a deal, I suppose. But what this suggests to us and reminds us is that whether molecules are in contact with each other, spatially separated, when they are in a single cavity and all cooperatively coupled to the same cavity mode, maybe we have the opportunity for energy transfer or some sort of communication or interaction between these separated slabs. And this non-local energy transfer has been demonstrated. This is very exciting, I think, demonstrated in excitonic systems with donor acceptor molecules, and very recently in the UCSD group's science publication, which provides some pretty strong evidence of cavity-mediated energy transfer between discrete molecules.
Hi, Blake.
Yeah.
Sorry to interrupt. We have one more question.
Mm-hmm.
Okay, is it question from me?
Yeah.
It's relevant to the discussion on the previous slide. So you said that you don't have any photons in the system. And I wonder, what about thermal radiation? It's always there. So it's deep enough so you can neglect it, or what?
I'm not sure. Yes, I've thought about this, but I don't know the answer. So there is black body radiation, there's thermal radiation and thermal photons bouncing around. Those are not, I believe, those are not going to influence the vacuum Rabi splitting or play a role in the strength of the interaction between the vacuum fields and the molecular oscillator. So even if you did, presumably, even if you did some reactivity experiments like the Evison group has been doing, and you were at, well, if you're at zero temperature, I guess that might impact the chemical reactivity, but I think without thermal photons, you would still see some impact of the coupling. Now, the fact that there are thermal photons might cause some other effects. Maybe under strong coupling, thermal radiation is altered. So the Evison group does have a paper about that, where they attempted to measure black body radiation. Well, they did measure the black body radiation from a strongly coupled system. But I guess what I would just say is, so maybe they play a role, yes, but they don't impact the coupling strength for the most part. So even if there are no photons present, there is still coupling.
Okay, thank you.
Okay. Okay, so this remote interaction is very exciting, and it's being seen and published on, so that's great. Okay, so that kind of highlighted some of our basic kind of zeroth order, first order work on cavity coupled systems and how we can use molecular and cavity properties to control coupling strengths and cavity molecular interactions. And so next I'll talk a bit about our electrochemical work. So the point here is that we, and I'm sure many other people, are often thinking about how, okay, we can see this spectroscopic response, we can see this splitting, and we can move the splitting a bit and control it and so on. How do we probe what an individual molecule is doing inside the cavity? You know, how do we, yeah, there are some spectroscopic approaches that might give us some information, but is there a somewhat independent way of interrogating individual molecules? And so maybe electrochemical probing might give us that. And so that's what we're trying to do here, is to design and build a system that will let us electrochemically interrogate reactions that go on within a cavity. So we put this system together. We have two mirrors that are gold. One of them is going to serve as a working electrode. And within this cavity, we'll load up a solution of redox active molecules. In this case, the redox couple is benzoquinone, dihydroquinone, Quinones are important for energy and charge transfer processes in biological systems and elsewhere. And in this form, in the oxidized form, the benzoquinone has a strong carbonyl, a strong absorber associated with a carbonyl band. So when we put a solution of these molecules, we can see strong coupling between a cavity mode and that carbonyl vibrational mode. Applying a reducing bias reduces all of those carbonyls and causes the splitting to disappear, simply because there are no more carbonyl oscillators to couple to the cavity. Okay. So you can basically switch the system between these coupled and uncoupled state. If you take a slice from those dispersion plots, it looks something like this. In blue are the two Polariton modes. And then when you reduce the system, you have a single cavity resonant mode there. Okay. We can examine this process a bit more closely. So this first plot here shows individual transmission spectra as a function of time under a reducing bias. And so initially you see basically in black 2 polaritonic peaks. There's an extra dip in that peak because the benzoquinone absorption has a doublet. But as you apply a reducing bias over time, and this is in this case minutes, those 2 peaks evolve to a single peak. There's also a rigid shift of the cavity mode. This is also an optical modulator. Let's see how good it is. So we can normalize the transmission to the initial state, and we see that at some frequencies, the transmission can be modulated 40 or 50 times, but very slowly. And right here, since this is a reversible redox couple, we can switch this system back and forth in and out of strong coupling. And we can even quantitatively extract the concentration of benzoquinone that remains at any given time point. Because we're collecting these spectra, we see the Rabi splitting, and we can fit various functions to those spectra. We also pull out the refractive index through that fit, and we see that there is a correlated change in the refractive index when we reduce the molecules. And this is important. So when this reaction happens, or when many other reactions happen, there is a change in the background refractive index of the solution. And that can be used to monitor reaction rate progress. And we'll see that in a bit. Since we have quantitative measure of the amount of benzoquinone remaining at any given time, we can do some rate analysis to see how quickly this bulk electrolysis reaction proceeds. It's fairly slow, scales linearly with the volume of material you have to reduce or oxidize. And if you extend this linear relationship down to a first order cavity, operational timescales are on the order of seconds. So it's still pretty slow as far as modulation goes. But remember, the point here is that we're creating a system that will allow us to use electrochemical tools and knowledge to monitor kinetics and overpotentials, et cetera, of reactions. And we're working on that now. And I have just one slide on another example of electrochemical switching. This is also the only system in the talk which is an excitonic strongly coupled system. And so in this example, we use a surface plasmon polariton optical mode on the surface of this gold film, strongly coupled to a methylene blue dye polymerized film. Methylene blue is a redox active dye, so we can, under its oxidized condition, it has a strong optical transition, and when it's reduced, that transition disappears. And so that switching is shown here. So in the oxidized state, there's a strong absorption around 650 or so, strongly coupled to the polariton, resulting in this splitting of the SPP. And then under reducing conditions, that splitting essentially disappears, and you just recover the dispersion of the SPP itself. And the spectra is shown here, showing that transition from strong coupling to no coupling. The spectra are a little complicated because the absorption features of the methylene blue are a little bit complicated. And again, though, this is a very flexible system and functional system, and it allows us to do very clean electrochemical measurements. These are a series of cyclic voltammograms taken at different scan rates. And if you plot the maximum current as a function of scan rate, you can extract the kinetics of this process. However, of course, as you might imagine, electrochemical kinetics are complicated and can be modified and altered by a number of processes. So we certainly can't say that we see any sort of strong coupling modified reaction kinetics now, but these are the types of things we're looking into. Okay. So now I will tell you about our first attempt at a modified addition reaction in a cavity. So we're going to look at this reaction right here. Cyclohexanol combines with phenyl isocyanate to form a urethane monomer. Now, full disclosure, coincidentally, this reaction is very similar to a reaction that is described in the supplemental of an archive manuscript by the Hiura group. So this is a group that includes a member of the Evison group who I think graduated and moved on elsewhere. So in their supplemental, they have a similar reaction. There's a different alcohol that's used. We chose this reaction based on this Nature Chem article here. So this comes from an ultrafast group who are pursuing coherent chemistry, I think is what they term it. And what they do is they've taken this reaction, these reactants, and with an ultrafast pump, prepared this reactant into the first vibrationally excited state. That prepared reactant then reacts at a different rate to form this product. And so they're looking at modifying reaction rates and reaction products by preparing the reactants in certain vibrationally excited conditions. Since there was a response that they saw that told us that this system is likely sensitive to the vibrational energetics, and so it should be a good candidate to use for such an experiment. So we did. And so for the out-of-cavity control experiment, we load these reactants between two transparent mirrors. We monitor the infrared absorption spectrum as a function of time, and we see that reactant bands like this isocyanate decay with time as that reactant is consumed. Other bands, such as this solvent, primarily a solvent band, remain relatively fixed with time, okay? And there's also a product band that grows with time, and that's associated with this carbonyl in the product. We can process this data, and from either the peak or the area of this isocyanate feature, extract quantitatively the remaining concentration of phenyl isocyanate, the remaining concentration of reactant with time. Since this is a second order reaction, we invert that, plot it versus time. The linear response gives you the reaction rate constant. So there's our out of cavity control rate constant. We then load the reactants into an optical cavity. So now we have two mirrors. The transmission spectra look quite a bit different now. So now the transmission spectra consist of a series of Fabry-Pérot fringes or resonant peaks. Depending on the cavity thickness, one of those peaks may be aligned with a vibrational band of interest. So in this example, we are strongly coupled to this phenyl isocyanate or the isocyanate vibrational band in that reactant. So when we collect a series of spectra like this, we now are tasked with pulling out the remaining concentration of reactant at every time point so that we can analyze that reaction rate. And the way the Evison group has primarily done this is by monitoring a higher order mode fringe and its movement with time. Because as the reaction progresses, the background refractive index shifts, and that fringe will shift accordingly. So we tried that, and this is our data here. So we look at a higher order mode. It shifts with time because the reaction is progressing. We can quantitatively correlate that shift in frequency with the background refractive index of the system because we know the cavity length L. We measure that before the reaction starts when the cavity is full of air. Okay. So now we know N as a function of time. Well, if we could only convert N to reactants concentration, we'd be in business. So let's see if we can do that. So one way we tried to do that was to look at a cavity that is tuned to that reactant, to the phenyl isocyanate. And so that we can extract the Rabi splitting, which is a proxy or proportional to the concentration of that reactant. So for every time point, we know what N is. We can extract this proxy for concentration, plot them versus one another. We get this linear relationship. That looks good. We should be able then to convert N to reactant concentration. But for us, so far, it hasn't been reliable. So we get data like this, but it's not consistent as we look at a couple of different cavities. Okay, so this, I mean, it doesn't wildly vary, but the conversion between refractive index and the concentration of phenyl isocyanate is not consistent. And this could be for a number of reasons. We could have solvent leakage or evaporation from the cavity, and that's going to shift these resonant positions, not because the background refractive index is changing, but because the mirror spacing can shift very slightly. Same thing can happen when the polymer components of your cell are not so robust. These are all exacerbated by the fact that our measurements for this particular reaction take place over hours or even a couple of 10 hours. Whereas most of the work, I think most of the Ebbison work is measured on the time scale of minutes. Okay. and lastly, when I pull out reaction rates from this data, they're noisy. And so my humanity kicks in and tells me, oh, there must be something wrong because the data doesn't look good. In any case, it is not particularly stable for us, and we think we know the reasons. It certainly can be a reliable method of monitoring reaction progress, given that these various processes are accounted for or ameliorated. And they're not nearly as important for short time frame measurements. So what we do in any case is a bit different. So I'm going to attempt to just measure the remaining concentration of phenyl isocyanate directly from the spectra. So first, we measure a series of absorption spectra of the phenyl isocyanate, okay, as a function of concentration. We plot the absorption coefficient as a function of concentration, and it follows Beer-Lambert law, of course, so it's linear. So I have a conversion between those two. Then we've made a series of cavities. Of course, some are tuned to that reactant mode, some are detuned from the reactant mode. But for all of them and for every time point, we collect an entire dispersion plot, an angle-dependent series of transmission spectra. So even for a detuned cavity, there's probably some angle at which a cavity fringe is tuned to this reactant. So for each of those cavities, for every data set, I look at the transmission spectra when we're strongly coupled to that reactant, and I fit that spectra to this expression. And this is the transmission through a single slab that has surfaces that have some reflectivity, so it's a cavity, and some spectrally dependent absorption coefficient alpha. So when I fit those spectra, I can pull out alpha, convert alpha to reactant concentration, invert it and plot it with time, and the data looks pretty good. And so again, my bias kicks in and I say, I must have done everything right because the data looks good. We know there are some problems though, however. This N in the expression here is a frequency dependent refractive index, but baked into that N is a fixed background refractive index. We know that that's changing with time, you know, consider the previous slide, but I'm assuming in this case that it's fixed and it equals the refractive index of the solvent. So this is something I'm trying to deal with. I would say we are still polishing what would be the best way of analyzing our data. But this is what we're doing so far, and this is what the reaction rates look like. Okay, so then let's look at the full data set. Okay, so again, here's the transmission spectra with the peaks labeled for reference. We look at a number of cavities. One, for instance, here has a fringe tuned to these solvent modes. Another cavity has a fringe tuned to this reactant mode. Another cavity is tuned to a product mode. And the last example here is a cavity that's detuned from all relevant modes. And so that's our uncoupled cavity control. The results are summarized over here. So again, the spectra are included for reference. This gray band in the center represents the out of cavity reaction rate constant. And the width of that band is the standard deviation of several measurements. Okay. The data points correspond to the reaction rates of various cavities, and they identify the cavity fringe positions, so you know how the cavity is tuned. The gray points are an untuned cavity. So that's basically this group of data here. And you see that when you carry out the reaction within a cavity, but those cavity fringes are detuned from any relevant mode, the reaction rate extracted is pretty close to the uncoupled control. So that's good. That's what you would think it might be. And then the colored data points correspond to cavities that are tuned in interesting ways. For instance, these guys are tuned to this batch of solvent modes, and they show a decreased reaction rate constant of maybe 40%, 40-50%. There are a few cavities represented over here tuned to a product mode, which also exhibit a rate decrease of 40%. Interestingly, when we look at the most prominent reactant peak, the isocyanate, when the cavity is tuned just to the red of that peak, there's little to no impact, maybe slight slowing. But when tuned more to the center or even a touch to the blue of that peak, we see an enhancement of the reaction rate. Again, maybe 40 or 50%. So overall, the data are showing cavity tuning dependent reaction rates. Now, unraveling exactly why and under what condition these modifications occur is tricky. Remember that these cavities have a number of different modes. And so finding cavities that align selectively with one mode and no others is not trivial. It's a challenge.
Hi, Blake.
Yeah.
We have one question from Joel.
Oh, well, I was waiting for your question.
Well, I mean, you inhibited me. I know. Well, I want to ask you a question. I mean, there is this very strange statement in the experiments by the Evesen group and co-workers, whereby the modifications are more prominent or mostly when the cavity mode at k equals 0 is the one that is strongly coupled. I mean, do you see that dependence that, say, if the strong coupling happens at different angles, do you still see these changes in the reaction rates?
Well, so that is, yeah, that's an excellent question. We think about that a lot and have, you know, no explanation essentially. But that touches upon, again, so imagine this. Okay, well, I'll say two things. Yes, if you imagine the dispersion of the system, my intuition would tell me that the strongest impact on chemical reactivity would occur when the optical mode is tuned to the red of the mode of interest. Because then when you look at the entire dispersion, it tunes blue, it sweeps through that vibrational mode, and you kind of maximize, if you will, the interaction between the optical mode and that vibrational mode. Okay, that's what my, kind of tinker toy mind might conjure. I guess we don't see that. If we just look at this somewhat limited set of data, we don't see that. But what I'm sorry, go ahead.
I'm a bit confused about that. I mean, I would expect naively that the strong coupling is pretty much independent on whether you are, the Rabi splitting is pretty much independent on whether you are at k equals 0 or whether you are at different k's. I mean, it might depend on mode profiles, but besides that.
Yeah, but you can have a mode. Let's say you had a mode that was slightly blue tuned from your resonance of interest.
Mm-hmm.
If you look at the entire dispersion, you might not have a fringe that interacts with that vibration at all, right?
Sure.
And so there, although a mode can be tuned at higher angle or at k equals 0, I guess the degree to which it's tuned, or the degree or the way that it can interact with the cavity, is still tuning dependent. That's why I suggested that if you were tuned just to the red of a given mode, you might be able to maximize, when looking at the dispersion, maximize the interaction between the cavity mode and that vibration, because you begin a little bit red from it, you sweep through it, you go a little bit blue, but are still maybe coupled a bit.
I see.
A larger portion of K space that's interacting with the molecule.
I see, but then you are saying that is not what you see. So then where do you see the strongest enhancement of the rate there?
Well, it looks like it occurs when it's fairly accurately tuned, or maybe even slightly blue, but I'm going to say that my data set is not so complete.
But when you say you say it's tuned at k equals 0, is that what
correct.
You mean?
That's correct. So all of these data points show where the cavities are tuned at k equals 0.
Okay, so then the point is that you also agree with the Evison experiments that there is a k dependence, that it does matter where the resonant mode is at a given angle. Like
it does matter. Yes, it does matter.
Okay.
Because, and think about this, I mean, if you really want to make your brain swim a little bit, let's just say, for argument's sake, that what really mattered, what really impacted the chemical reactivity, is what mode was tuned at 45 degrees. Let's just say, for argument's sake, it was some higher K that really made all the difference. Well, then all of these data points on my plot, you would sweep them blue, several hundred wave numbers, and maybe get an entirely different, picture of the interaction. I'm choosing to plot where the resonances are at k equals 0.
Sure.
You could plot maybe an entire, make each point very wide so that it covered the entire dispersion. Mm-hmm. But it would end up not communicating much. But at this point, we don't know. We don't know why it seems to be important that it happens at k equals 0, but that seems to be the case.
Okay, thank you very much.
Okay.
Hi,
and so let's
Rick.
Oh yeah.
We have one more question from Wei.
Sure.
I have two questions. One is there seems to be also a enhanced reaction activity at a frequency that is redshifted from the solvent mode.
Yes. So let me explain that. Yeah, I didn't mention this. So so recall that all of these cavities have multiple resonances. So for instance, this cavity associated with this blue point has multiple fringes. I've decided to indicate the position of the fringes that I thought were relevant. So there's a fringe here, and that blue cavity also has a fringe over here. There's probably one or two other fringes in here, but I didn't indicate them because they're not aligned with anything.
Right.
The same for this orange data point. So all of the cavity, all of the fringes associated with a single cavity have the same color. So for this orange labeled cavity, there's a fringe right here, which is kind of right next to the phenyl isocyanate. There's another fringe that's kind of close to these solvent modes that you're asking about. So honestly, I don't know why the reaction rate is faster. I don't know why the reaction rate is faster for that cavity. Is it because we're coupled to the isocyanate reactant, or is it because we're tuned just red of this batch of solvent modes, and that's what's making all the difference? We don't know. But I thought that it was close enough to these modes to indicate that there is a fringe there.
Sure.
Yeah.
Great. Thank you. I have another question. So what's the width of your cavity mode? Because it seems like when you couple it to the reactance mode, it's very sensitive to the detuning.
Yeah, that's important. So the width of the cavity mode is kind of going to determine the resolution with which you can make such plots.
Right.
Roughly, the width of the cavity modes are the width of these dots. So I think it's like 10 to 30 wave numbers.
Mm-hmm.
Yeah.
Thank you.
Okay. Okay, so we'll look just a little bit closer at this data to see, so you can judge for yourself how convincing it is. So I've put here some examples of the data from which I extract the reaction rates, and they're color coded to match the plot points. And so you see up here these two enhanced reaction rates in orange and yellow. The orange data certainly exhibit a bit of scatter. The yellow is much better behaved. And then here are the slowed reaction rates in this purple and blue and so on. And basically the point here is for a viewer, you all, or reader to kind of evaluate how significant the reaction rate modifications are, and are they much larger than the noise associated with these linear fits? And I would say yes, I think. And then lastly, if you're paying attention maybe and thinking deeply about it, you might say, okay, fine, but wait a minute, Blake, if you've got a reaction rate modification due to cavity coupling to this isocyanate band, for instance, as that band is consumed with time, coupling strength should decrease, and that should probably impact the reaction rate or the modification of the reaction rate. So let's look at that. So here is the reaction rate as a function of time. Again, this gray band is the out of cavity response. And so we see that the enhanced reaction rates over time do tend to decay to this uncoupled rate. This gray data is the uncoupled cavity mode. And although it increases with time, it roughly remains within this band of the uncoupled or the out of cavity control. So that's good. These slowed rates down here don't seem to change too much with time, but let's consider, remember that they are associated with cavities coupled to this batch of solvent modes, which don't change with time. There is one cavity coupled to a product mode, which should grow in with time and its influence becomes stronger. Although I will say that because we collect a full dispersion at every time point, our time points are very coarse. And within the second time point, there's enough product to be strongly coupled to the cavity. So maybe that's an explanation, I'm not sure. But overall, I think we show cavity tuning dependent reactivity. There is a correlation between reaction rates and cavity coupling to specific vibrational modes, and that qualitatively the time-dependent behavior of those reaction rates is roughly what you would expect. Okay. And I'm 5 minutes over, so I'll say, let's see if I can do 4 words on each slide. Dunkelberger measured key transient features. That's 5 words, sorry. We teamed up with Wei and Hoel's group at UCSD to do some 2D measurements. We have since built up our own 2D IR system and have recently made some unique observations in a strongly coupled system. And with the help of Felipe Herrera and his crew at University of Santiago, identified those as polariton excited states and extracted their kinetics. And so pretty soon here, you should be able to read that whole story in more detail. And that's it. So that's it. So we basically tried to start with the basics of understanding how cavity coupling and Rabi splitting and other system observables depend on system properties, and then how to translate our understanding of those phenomena to interpret chemically reactive systems within cavities. And that's what we're moving toward. And we get most, I guess, all of our money from the Navy from one way or another. And lastly, for all of those people who missed the deadline for the other invited issue, in a number of months from now, I guess this is probably 6 or 8 months from now, I'm helping to organize an invited or a special issue of Nanomaterials, which is a reasonably impactful journal. And this special issue is on nanophotonics for light matter interaction, not necessarily focused on chemistry, but on how various characters of the photonic modes affect light matter interaction. So feel free to contact me or Google that up and see if it's of interest to you. And that's it. I'll take any more questions. Thanks a lot.
Thanks, Blake, for this wonderful talk. So we do have several questions, and it's time. So maybe Hoel.
Great, Blake, that was very interesting. So a quick follow-up. So I think we have talked about this, but were you able to test whether just weak coupling makes any difference in the kinetics?
Yeah, so that's a good question. So I think the concept in general is that it may be difficult to describe everything that coupling imparts, but surely coupling some mode to a cavity will introduce a loss channel, and weak coupling should also do that. I will admit here that the plethora of cavity modes that we have in this system include a couple more that interact weakly with other reactant and product modes. Okay, and so just as I was telling Wei, for instance, for this orange cavity, we see an enhancement of the reaction rate. Is it because the cavity is coupled to this isocyanate mode? Is it because it's slightly red tuned of this solvent mode? Well, I'm not really sure. We convolute the interpretation further if we start to include weakly coupled modes. And
but can you not reduce the concentration enough so that you don't see peak splittings?
That's true. That's true. So we can do that. We have not done that, but we should be able to do that.
That would be interesting to see.
Yeah. So that would be a bit more difficult just because of the way that we measure the reaction progress. We kind of rely on strong coupling and having a very prominent spectrum with splitting and other features that we can fit to. If we reduce the concentration such that we were weakly coupled, we might have to rely on the refractive index method, and in that case we'd have to kind of tighten up our approach a little bit so that it was more reliable.
Thank you.
Mm-hmm.
And questions from Marisa?
Hey, thank you so much, Blake. It was a really beautiful, clarifying talk. I have a question thinking about these different cavity modes that you're not sensitive to in your measurements. And I wonder if you've thought at all about transverse modes and whether there are vacuum transverse modes that could be doing coupling that you're not observing.
Yes, so there is, I can't comment very intelligently about that. But yes, they do exist. I suppose there are also modes in free space. So this is where your mind starts to go. There are also modes in free space. And so when someone does a reaction in a beaker, are vacuum modes within that beaker, you know, playing a role in reactivity? And I don't know. And so if there are transverse modes inside this cavity that are having an impact, I don't see them, of course. And if they're having an impact on some property, relaxation, or reactivity or something like that, we can't really say anything about them. I guess what you would try to do is just design a system that had them and design a system that didn't have them, and then look and see if there's a difference. That's kind of the game, I guess, you have to play.
Yeah, okay, thanks.
Another question from Bill Barnes. Hi, Bill.
Hello. Blake, thank you. Very nice talk. Really interesting stuff.
Thanks.
Very nice to see a lot of that. I wanted to just ask you very briefly if you could say a few more words about what came right at the end, the excited state Polariton stuff. Can you just describe very briefly what the topic was? Because I'm fascinated by that.
Yes. So my collaborator Jeff Horutsky gave a full talk on this in this series, but it was several months ago, and so I'm not sure if I remember exactly where we were status wise at that time. But essentially, and I'm going to ask Hoel and any sort of theorist to avert their eyes because I think this picture gives them heartburn. But if you have a ground state, first excited state, and then a couple of polariton states, right? This is a standard picture. And then you pumped, and we know what the transmission spectrum of this looks like. And if you pump the system, well, you can move population around. And early on, we figured out quickly what the excited state spectra looked like if you just move population to the first excited state of the molecule. Okay, that was pretty straightforward. But understanding what the excited state transmission spectrum looked like when you had some sort of coherent population in the lower or the upper polariton was less clear. We tried to draw some admittedly half-assed pictures to try and explain what we saw early on, but there was really very little rigor behind it. However, recently, and I think there may be a number of theoretical descriptions that do adequate jobs. To be honest, I'm not fully familiar with all of them, all of the theoretical work. But Felipe's model, which is described in this paper right here, I think this chemical physics paper, allows one to predict what sort of excited states are accessible to those coherent polaritonic excitations, okay? And so in this latest work that we've done, we've tried to look at the very early times of the transient response where these coherent polaritons remain alive. And the features we see seem to coincide pretty well with his predicted polaritonic excited state transitions in both frequency position and in magnitude. So he's able to extract some transition dipole moments of them. And And then once we are able to say, okay, this feature here is a polariton, is a lower polariton transition to some upper polariton, upper middle branch, or an upper lower polariton, something like that, then we can pull out the kinetics of it. And that's what we've done here. And if you want, I can, send you the preprint. I think we're doing proofs now on that. So it's pretty much wrapped up.
That would be nice, but I have to say that the half-assed picture at the beginning was the bit I really wanted. so thank you very much for that. Really nice. Thank you.
And one last question from George.
Hello.
Hi.
So thank you for this very great talk. I just have a very quick question. Have you been able to compare the results for rates that you get with both kinds of measurements? So from refractive index and your second method?
That was my goal. That was absolutely what I was going to try to do. We had done a full analysis using this frequency shift method, and it looked pretty good. Again, my human bias was kicking in. There were modifications when the system was tuned to reactant modes, et cetera. And I said, okay, well, this is great. Well, but we also have all of this other complementary data that has the Rabi splitting right there. So we can directly extract the concentration. Let me just analyze 1 or 2 sample sets that way. It should give consistent results. And that would be a nice contribution, a nice part of this paper. But it was different. And so that caused me to dig into it a little bit more. And that's where I found for our measurement conditions, for these very long measurements, that the frequency shifting approach was not consistent. Yeah, and so that's where that left us. Yeah. But again, I want to emphasize that for shorter time scale measurements and for first order kinetics rather than second order, it's much easier to use that shift frequency shift approach, and it's completely legitimate. Yeah, so there you go.
Thank you.
Yeah.
Okay, I think that's all. And thanks, Blake, for this wonderful talk. And next week we'll have this same Polariton webinar at the same time, and the future advertisements will be posted later. And that's all for today. And bye.
Thanks, everyone. Bye.